Hello students, welcome again to the MicroMed education channel, and today we'll discuss regarding the history of microbiology. The first important thing one should have in your mind why we need to learn the history of microbiology and whether it is important for our exam point of view or for NEET PG exam or university. We have to understand that a microbiology subject, the history is very important because we need to know various discoveries done by which scientist, what they have discovered. This is frequently asked as a short note as well as in 2 marks question as well as in MCQs of NEET PG exams. So let's begin our today's topic. So in the beginning, before 1600, previously people thought that the disease are caused by a curse, and after the curiosity of the scientists, it was identified that the living agents are related with the disease formations, and they are later on detected by the microscopes. So in 1600 to 1800 era, the first discovery was done by Sir Anthony van Leeuwenhoek. What he discovered with observe the microbacteria, that is bacteria, using a single hand microscope. Then he called this as animalcules. After that, Edward Jenner first developed a vaccine, that is a smallpox vaccine, but it was prepared with the use of a cowpox virus. Then the later on the term vaccine is evolved. In 1815 to 1900, Sir Louis Pasteur, who is also considered as father of microbiology or father of bacteria. Actually, the father of microbiology is considered Sir Leeuwenhoek because Antony von Leeuwenhoek, because he first discovered the microscope and later on the microorganism started observing. But Louis Pasteur को भी father of microbiology और especially father of bacteriology कहा जाता है. And he discovered spontaneous generation of theory like germ theory, etc. Then comes Joseph Lister, that is also known as the father of antiseptic surgery because he used carbolic acid to sterilize instrument. Robert Koch, he discovered various bacteria like anthrax and cholera and also proposed Koch's postulate. In 1900 onwards, Paul Ehrlich, it is also called as father of chemotherapy because he discovered one special drug that is called salvarsan for syphilis. Alexander discovered an antibiotic with the discovery of penicillin from Penicillium fungus. DNA was discovered by Watson and Crick, and that is how a molecular microbiology has started blooming. Then comes Carlsworth and CRISPR technology. So this is the summary timeline. We'll revise that in 1600. First of all, Sir Leeuwenhoek, who is also considered as father of microbiology. Yes, he discovered microscope. In 1700, Edward Jenner, he developed first vaccine. In 1800, germ theory by Louis Pasteur and Joseph Lister proposed the theory of asepsis. In 1900, chemotherapy by Paul Ehrlich and antibiotic by Sir Alexander Fleming. In 2000 onwards, molecular microbiology. Now we'll begin with individual scientists and we'll use certain mnemonics to remember their discoveries, so it will be helpful. Now which short notes will be asked? The first is Louis Pasteur, Robert Koch and Paul Ehrlich. We'll first discuss with the Louis Pasteur how you can remember their discovery. So remember this mnemonic: fresh milk and rabbit get germ-free life. How this we'll discuss just now. So Sir Louis Pasteur, he invented various things. So how you can remember? Fresh milk and rabbits get germ-free life. F, that is fresh for fermentation. He proposed principle of fermentation. M for milk. Pasteurization milk was discovered and developed by him. The word is develop is better than the discover. Then he developed anthrax vaccine. Remember, the anthrax bacilli was discovered by Robert Koch, but anthrax vaccine is developed by Louis Pasteur. Then comes, this is fresh milk and ant. Now rabbits, he developed rabies vaccine. He proposed germ theory and disapproved spontaneous generation theory, and germ-free life, that is the principle of sterilization. He introduced various sterilization technique. So in this manner, you can remember all the discoveries produced by Louis Pasteur. Then comes Robert Koch. The mnemonic here is Koch's lab acts purely. So L stands for, he introduced liquid media and agar media. A, as discussed earlier, he discovered anthrax bacilli, and Louis Pasteur developed anthrax vaccine. He also discovered tuberculous bacilli and cholera bacilli. So basically, Sir Robert Koch discovered three bacteria. Tuberculous bacteria is also called as Koch's bacilli, that's why. Cholera bacteria and anthrax bacteria. He proposed and introduced various aniline dyes for various staining techniques. Developed various pure culture techniques with the use of culture medias. He proposed Koch's phenomena, that is a tuberculosis test that we will discuss just now in a while, and also the Koch's postulate, which is scientific proof and development of petri. So Koch's lab acts purely like L for liquid solid media, A stands for anthrax bacillus, tuberculous bacillus and cholera bacillus. Propose or introduce aniline dyes for staining, C for culture technique, 2 tuberculosis basically for Koch's postulate, and then Koch's phenomena, T for Koch's phenomena by tuberculosis test, and S for scientific for Koch's postulate and petri. Now we'll discuss what is Koch's postulate. In this, a sick animals proves again. Now how this just now we'll discuss. Understand that whenever any person get disease, the microorganism is constantly present in that particular person. So seen in every case, when you can always isolate that particular microorganism from this patient in a pure culture, that is I. Then see, if you use this pure cultural isolate and introduce in a healthy person, it also causes the same disease. And again from the same patient, you can again isolate that organism in a petri dish or culture media. And later on, the fifth postulate is introduced that antibodies to that particular causative organism is present in that diseased person. So we'll revise again that a microorganism should be constantly present. Patient के पास जिसको जो disease है उसमें वो microorganism constantly present होता है. वो माइक्रो ऑर्गेनिज्म को उस पेशेंट के सैंपल में से प्योर कल्चर में ग्रो कर सकते हैं. उसी कल्चर एक्सट्रैक्ट को अगर किसी हेल्दी इंडिविजुअल में इंट्रोड्यूस करें तो उसमें सेम डिजीज प्रेजेंट होता है और उसी पेशेंट में से फिर से वो प्योर ऑर्गेनिज्म को हम कल्चर में ग्रो कर सकते हैं. एंड फिफ्थ इज दैट द सेम पर्सन इन विच द डिजीज वॉज प्रेजेंट, उसके अगेंस्ट उस ऑर्गेनिज्म के अगेंस्ट की एंटीबॉडीज आर डेवलप होती है. सो दिस इज कॉल्ड Koch's postulate. Now we'll discuss what is Koch's phenomena. First we'll understand, then we'll understand the mnemonics. इसमें क्या हुआ था कि Koch has injected a guinea pig with a tuberculous bacilli which he has also isolated and discovered. So then after some time he re-injected the mice with the same tuberculous bacilli, and after 24 to 48 hours the site where again it was injected, it has formation of tuberculoma, that is something called a delayed hypersensitivity reaction. So the mnemonic is fat pigs reacts fast. F, that is first infection where the immunity is sensitized with that particular tuberculous bacilli. A, again introduced, leads to tuberculin reaction, that is local necrosis and ulceration, that proves the patient has immune memory, that is called type 4 hypersensitivity reaction, that is a fast response that occurs in 24 to 48 hours, which is delayed type of hypersensitivity reaction. Now we'll Move to our another scientist, that is Paul Ehrlich. Now, what this Paul Ehrlich discovery will understand. So Ehrlich provided the magic site saved immunity. Now remember by this mnemonic his various discoveries. So first of all is Ehrlich phenomena. He used toxin and antitoxin interaction and proposed the basis of immunity. He introduced various histological stains, E and H as already discussed. Now R. R is receptor, that is side chain theory. He proposed side chain theory for the formation of antibody. Then he proposed the lytic, that is immunity concept, and he is considered as the pioneer concept of humoral immunity. He is the founder of Paul Ehrlich Institute in Germany. He proposed chemotherapy. That's why he is also known as father of chemotherapy. Then the magic bullet drug that he discovered is salvicillin, that is used for the treatment of syphilis. That is his magic site has saved. So this is the mnemonic Ehrlich's magic site saved immunity. By this mnemonic you can remember all his discoveries. So the take-home message of our today's lecture will briefly revise and remember. Father of microbiology, Sir Anthony van Leeuwenhoek, who discovered microscope. Father of bacteriology, and some people also call as father of microbiology, Louis Pasteur, various discoveries by them. Father of immunology is Edward Jenner, who developed vaccine of smallpox. शीतड़ानी वैक्सीन जे कहिए शीतलामा, जो previously उसे ऐसा भी बोला जाता था कर्स. Later on it was understood it is by a virus infection. So Edward Jenner developed a smallpox vaccine using cowpox virus. Then father of antiseptics is Joseph Lister. He used carbolic acid and introduced the subject of asepsis and antisepsis. Then father of chemotherapy is Paul Ehrlich. Thank you so much. Hope this find you interesting as well as easy to remember. You can get this same PDF copy in the link that is present in the video. You will get a link of the Google Drive from where you can download the notes for the history of microbiology. Thank you so much, and please subscribe my channel, and will regularly update various topics with the videos and PDF formats. Thank you so much.